Hello guys and welcome to my channel The Bio Guru, where science becomes sick. Today's lesson will be glycolysis, which is one of the fundamental topics in CIE A2 biology. So without further ado, let's dive into today's lesson. Before starting the lesson, I want you all to know what glycolysis means exactly. Because in biology, certain terms are easier to understand and remember if you know what they actually mean. And glycolysis is one such coined word which has its literal meaning embedded in it. So let's see what the term glycolysis means. In biology, the term glyco usually refers to a sugar-associated compound. And in this case, the compound it's referring to is glucose. Meanwhile, lysis means the splitting or breaking of a substance. So as the name suggests, in very basic terms, glycolysis is the splitting or breaking down of glucose. So glycolysis begins with the 6-carbon molecule we call glucose. Now in this case, I want you to think of each circle as a single carbon atom because I have drawn 6 circles, all of them resembling the 6 carbon atoms of glucose. So the 6-carbon glucose molecule is phosphorylated using ATP or adenosine triphosphate. Now, what is phosphorylation exactly? Phosphorylation is the addition of one or more phosphate groups to a compound, and in this case, two phosphate groups are added to the first and sixth carbon atoms of glucose to form a phosphorylated glucose molecule which we term as fructose 1,6-bisphosphate. Now you would have also noticed that I have said phosphorylation using ATP. Now that is because ATP acts as a source of the two phosphate groups which we use to phosphorylate glucose. Because ATP is a phosphorylated nucleotide with 3 phosphate groups. So when you take two such ATP molecules and hydrolyze them to adenosine diphosphate, then the inorganic phosphates are taken and added to the first and sixth carbon atoms of glucose to form fructose 1,6-bisphosphate. So in short, I can say two ATP molecules are used to phosphorylate glucose. Why is phosphorylation necessary? Now, if you consider glucose on its own or in its ground state, then it is generally or relatively an unreactive compound. But in this case, we need glucose to be highly reactive so that it can be split into several compounds before forming the ultimate product. And this would not be possible if glucose was a very stable molecule. Therefore, in order to make glucose more reactive or to lower the activation energy of the reaction, we convert it to a more unstable or a more reactive form called fructose 1,6-bisphosphate. Now you would have noticed that I have written this 1 and 6 within brackets. That is because at CIE A2 level, you are not expected to write 1,6 in your answer sheets because it is not specified in most of the mark schemes. However, it is good to know what it means. So it's optional and you can decide whether or not to include it. After the phosphorylation of glucose has taken place, this molecule, which is termed as fructose 1,6-bisphosphate, is split in a process called lysis into two 3-carbon triose phosphate molecules. Now, as I mentioned previously, lysis in biology means the splitting of or breaking down. So in this case, fructose 1,6-bisphosphate is broken down into two 3-carbon triose phosphate molecules. I want you to know that these silver colored circles represent the two phosphate groups which were initially used to phosphorylate glucose. After lysis has occurred and triose phosphate molecules have been formed, these triose phosphate or TP molecules in short undergo a series of many intermediate reactions during which intermediate compounds are formed, and ultimately the final compound, which is pyruvate, is formed. So from two 3-carbon TP molecules, we get two 3-carbon pyruvate molecules after a series of intermediate reactions. Now I have just said many intermediate reactions because at CIE A2 level you are not expected to know the exact terminology of the reactions in between or the names of the intermediate compounds formed. So it would suffice if you know that there are many intermediate reactions that cause the conversion of TP molecules to pyruvate. Now during these intermediate reactions, there are two vital things that occur. Number one is ATP synthesis, which I will be discussing in a few moments. And number two, as mentioned here, is dehydrogenation. So during these reactions, dehydrogenation occurs, meaning, as the name suggests, dehydrogenation is the removal or elimination of hydrogen from a compound, and most probably from an intermediate compound in this case. So the hydrogen released cannot be left on its own. Therefore, we have a coenzyme called NAD. You do not need to know what NAD stands for. You just need to know that it's a coenzyme. So this NAD coenzyme, also known as NAD plus in its symbolic form, accepts hydrogen, and if you are learning chemistry, you will be knowing that gain of hydrogen is called reduction. And so when NAD plus or coenzyme NAD accepts hydrogen, it becomes reduced and forms reduced NAD, whose symbolic form is NADH. Now, per glucose molecule, there are two reduced NAD molecules formed. Per glucose molecule, there are two reduced NAD molecules formed. Now let's move on to how ATP is synthesized during glycolysis. So as I mentioned before, during the many intermediate reactions that cause the conversion of triose phosphate to pyruvate, ATP is synthesized. Now during glycolysis, ATP is synthesized by a special process known as substrate-linked phosphorylation, and sometimes you will also come across this term in a different name called substrate level phosphorylation. Both of these names, substrate linked and substrate level, mean the same thing. So now let's have a look at what exactly substrate linked phosphorylation is. Phosphorylation, as you know by now, is the addition of one or more phosphate groups to a compound. And in this case, to produce ATP, we are phosphorylating ADP or adenosine diphosphate. And this term substrate linked suggests that there is an involvement of a substrate to produce ATP. So to attach a phosphate group to adenosine diphosphate, we need energy. And this energy is caused by the splitting of glucose, or basically the breaking of bonds between the carbon atoms of glucose. Therefore, in simple terms, we can say substrate-linked phosphorylation is the addition of a phosphate group to ADP, or adenosine diphosphate, to form ATP, or adenosine triphosphate, utilizing the energy released from the splitting of a substrate, which in this case is the starting compound glucose. So now that you are hopefully clear with what substrate-linked phosphorylation is, let's take a look at the general overview of the amount of ATP produced. Now, as I mentioned at the beginning, during the initial part of glycolysis, two ATP molecules are used to phosphorylate glucose into fructose 1,6 bisphosphate. Therefore, ATP used during glycolysis is equal to 2. And by substrate-linked phosphorylation, at the end of glycolysis, 4 ATP molecules are produced. This is the gross production of ATP, and the gross production of ATP is 4. However, since 2 molecules are already used at the beginning, in order to calculate the overall manufacture or the net manufacture of ATP, we need to subtract the total amount used from the total amount produced, meaning the net production of ATP is equal to 4 minus 2, which is 2. Therefore, simply we can say glycolysis produces a net or overall value of 2 ATP molecules. So that brings us to the end of today's lesson. Hopefully you all have now fully understood, or at least have a better understanding of glycolysis than you did before watching the video. If you enjoyed today's lesson, please like, share and subscribe, and don't forget to drop your comments down in the comment section. Thank you all and see you soon in our next lesson.